The logistics, exactly the same. Okay, so your lecture exam will have 50 questions, of which 40 of the first ones are multiple choice, and 10 at the end are fill in. Okay, and you have 50 minutes unless you qualify for extended test taking time. And if you do qualify for extended test taking time, make sure that the Canvas has denoted that you have the extended test taking time. If you don't see that, email me right away and I'll see what I can do to change the settings. Okay, but you should see the extended test taking time for that. So the lecture, 50 questions, 40 multiple choice, 10 fill in the blank. And like always, I divide them evenly. So the cardiovascular unit has 3 chapters, and then respiratory has 1, digestive has 1. So there's going to be about 10 questions per chapter. Okay. And then since the blood vessel is a smaller chapter, you might see a little bit shift of questions in there, but it's usually 10, 10, 10, 10, 10. So 10 each because there's 5 chapters in this unit. Okay. Any questions about the lecture exam? Okay. So it is available from Sunday to Monday. Make sure you do finish submitting it by 11 p.m. Central Standard Time. You will need to have for both the lecture and the lab exams the Respondus Lockdown browser. Down. Plus the webcam. And it will ask you that when you're using the webcam to do an environmental scan. And again, that's been really important. And it's more for, for you in case Respondus Lockdown flags you and I have to watch. The video, that's the very first thing I go to is the environmental scan to make sure that there's nothing on the right side of your table and the left side of your table and in front of you and behind you, okay? So if you have any questions about how to do the environmental scan, I did provide an example of what one would look like. Okay. All right, I think that's it for the lecture. And then for the lab, same thing. There's going to be 30 questions, and it is all identification. Spelling counts. And now, in this unit, you notice that you have a lot of right and lefts. So if there's a pair of something, make sure you do indicate whether it's the right or left. If it's a blood vessel, make sure you indicate whether it's an artery, vein. So however you see the name in the PDF, the lab PDF, make sure that that's how it shows up on what you type, okay? So let's say if a term has 3 parts to the name of that structure, make sure you include all 3 parts, okay? And your lab is going to be on the heart, the lungs, so essentially the respiratory, all the organs that you had to know for the respiratory, and then the digestive as well. So there's about 10 questions on the heart, 10 questions on respiratory, and 10 questions on digestive. Okay, and same thing for the lab as is for the lecture. It will require Respondus Lockdown, webcam with environmental scan. Any questions? Okay, I think you're pros at this now, so we'll just go right into it. So cardiovascular is called the cardiovascular because it includes blood, heart, and the blood vessels. Okay, and we'll start off with blood. So for the blood chapter, make sure you know the function of blood. And someone just remind me, what are some of the functions of blood? We're such a small group. You can feel free to leave yourself unmuted.
Is it protection, regulation, and transportation?
Yes. One, the main one is transportation. Okay. Think of blood as, it's the fluid that moves everything throughout your body. Okay. So it transports everything that needs to go throughout your body, like the waste, nutrients, gases, everything travels through blood. Okay. So transportation is one of the big ones for sure. It also regulates. And what does it regulate?
pH.
Yes. What else?
Body temperature.
It could, but you'll find that blood
Oh,
carry the, yeah, go ahead.
isn't it blood pressure?
Yes, that's what I'm looking for.
Okay.
And the reason why it regulates blood pressure is because blood pressure is based on what?
Water.
Yes. So if blood has a lot of water in it, the blood pressure is going to go up. And if there's less water in blood, then the blood pressure goes down. So because it carries water, the amount of water it carries, of course, is going to influence the blood pressure. Okay? And so on. So it regulates a lot of levels like blood pressure, pH, blood volume, and so on. Okay? What else? One more function of blood.
Protection.
Yes. How is blood protective?
Well, it carries the sites, the cells that needs to go ahead and like the white cells
That's it.
through it delivers them to the muscles and everything else.
Exactly. So it carries the white blood cells, which are your immune cells, right? And that's how it's protective. It's protective in the sense of immunity. It also carries platelets, which is a clotting factor. And one more that's also a clotting factor. What would that be?
It's a type of white blood cell, right?
Nope.
No, can't remember.
You have two clotting factors in blood. One of them is platelets. What's the other one? Who can remember?
Is it the fibrinogen?
That's absolutely right. So it's the plasma protein. And specifically, it's the fibrinogen. That's also a clotting factor. Very good. In addition to that, so not only do you have the white blood cells, you also have the clotting factors, but you also have another plasma protein that functions in recognizing what should be in your body and what shouldn't be in your body. And oftentimes that's referred to as self versus non-self. So what is it looking for? Which plasma protein am I talking about? I'll give you a
You're
clue.
talking
Yeah,
the
go ahead.
albumin?
So close. I think you mean globulin, right?
Oh, I was saying the other one, but okay, globulin. Okay.
Yes. So there's a plasma protein called globulin. And then there's three types, right? There's alpha, beta, and gamma. And it's the gamma one specifically. That's the immunoglobulin.
Oh, okay.
And essentially, these are antibodies. And they also protect you in that they can identify what is you from what's not you. Okay, so they would bind to antigens that are non-self. Okay, so that's antibodies. So that's how they protect you. So when we say that blood is protective, it's protective in three ways. One, You've got the white blood cells that are your defenders. It like directly destroys things that shouldn't be in your body. It protects you by, if you were to bleed, it helps you to stop that bleeding through clotting factors. And it protects you in identifying, being able to distinguish what should be in your body and what shouldn't be in your body in the form of antibodies. Okay? All right. Then when we look at components of blood, we do what's called a hematocrit. And a hematocrit essentially is they would draw your blood, put it into a tube, and put the tube into this machine called a centrifuge. And the centrifuge spins the tube. And when it spins that tube, the components of blood is going to separate based on density. The most dense goes on the bottom, the least dense goes on the top. So really, when you look at the components of blood, there are three main divisions. What are they? Let's start with the least dense. So what would be on the top? Makes up about 55% of blood.
Plasma.
Plasma.
Perfect. Plasma. So plasma is the fluid of blood, and that's why it's the least dense. Okay. And in the middle, you will find what? Less than 1%.
Water.
No,
Buffy coat.
the buffy coat. Perfect. Now, someone has said water, and water is actually in plasma. So plasma is like 80 to 90% water. So that's where water would fall into, is in the plasma. Okay. The 1%, the less than 1% is the buffy coat, and the buffy coat consists of two parts. What are they?
Leukocytes.
Yep. So the white blood cells, which are also known as leukocytes. And one more.
Platelets?
Platelets.
That's perfect. True or false? Platelets are cells.
False.
Beautiful. So platelets are clotting factors, right? And they used to be called thrombocytes. Thrombo because they clot, cyte because we thought that they were a cell. But they are not cells. They're actually fragments of a larger cell called a megakaryocyte. So because of that, we no longer call them thrombocytes because we know that they are not cells. They're fragments of a larger cell called a megakaryocyte. Okay? So platelets are not cells, but they are factors that allow for clotting. Okay? And they are part of the buffy coat. Now, it makes sense that the buffy coat makes less than 1%. Because in a healthy person, do you need a lot of white blood cells?
No.
No.
And if you're not bleeding, would you need a lot of platelets?
No.
No.
No. And that's why in a healthy person, it's less than 1%. So if you feel sick, you go to the hospital, they'll take your blood. And if they notice that your white blood cells are elevated, that's a bad sign, right?
Yes, because that means you're sick.
That's right. That means that you're trying to fight off something. And then they'll look even closer and look at what specific white blood cells elevated, because different white blood cells respond to different infections. And we're going to learn about that in a little bit later. Okay, but for now, the buffy coat consists of white blood cells and platelets, makes less than 1%. Then the other component of blood makes up 45% of blood. What is that?
The red blood cells.
That's right. What's another name for red blood cells?
Erythrocytes.
Perfect. Erythrocytes. Wonderful. Okay, so that's the components of blood. So you have the plasma, the white blood cell, the platelets, and the red blood cells. Okay, so that's the components of blood. Now, the components of blood can be divided into two categories. One category is referred to as formed elements, versus non-formed elements. So what of the component would be considered formed elements? In other words, what's a cell? What has structure to it? For sure your red blood cells, for sure your white blood cells, and then the platelets, because they come from larger cells. So the non-formed elements of blood would be plasma. Okay, so that's just categorizing the components of blood. All right, so now I want to move in and talk about red blood cells because there was a question about it as well. Okay, so red blood cells are also known as erythrocytes. Make sure you understand the function of red blood cells, the structure of red blood cells, and the life cycle of red blood cells. Okay, so let's start with the function. What are some functions of red blood cells?
They transport the hemoglobin that carries the oxygen with it.
So they transport gases. And what particular gases?
Oxygen?
Yep, and one more.
Carbon dioxide.
Very good. Okay, so they transport oxygen and carbon dioxide. Okay, all right. So in terms of structure, they're going to look something like this. If you're looking at the top view, it looks like a donut. If you're looking at the side view, it looks like this. Okay, so overall, what term do we use to describe the shape of a red blood cell? So because it's round, we often use the term what? Discoid, because it's round, it looks like a disc. We also use the term biconcave because if you look, it dips in on the top and on the bottom. So it's referred to as biconcave. Why do you think it's biconcave?
Because it's caved in on the sides.
Yes, but why? What would you normally find on the middle of a cell?
Oh. um,
Nucleus?
Yes, the
Yeah.
nucleus. So since it's biconcave, it tells you that a mature red blood cell is what's wrong with it. It's not so much what's wrong with it, but what is it missing?
it's missing that because it doesn't have a nucleus or a mitochondria.
That's right. So it's biconcave because it has no nucleus. Because it has no nucleus, and you know that a nucleus contains the genetic information, right? So because it has no nucleus, it doesn't have the genetic information, it can't repair itself. So because it has no nucleus, it can't repair itself. It has what type of lifespan, short or long?
Short.
Yeah, and approximately about how many days?
120.
Yeah. So a red blood cell only lasts for about 120 days. Why? It doesn't have a nucleus. And that's why it's biconcave. Okay, and because it doesn't have a nucleus, it can't repair itself. So hence 120 days. Okay, but that's okay. It's not a problem because why? You are constantly making new red blood cells. Okay, and like you had said, it also has no mitochondria. Makes absolute sense.
So how does it get its energy?
Yeah, that's a great question. So mitochondria facilitates aerobic respiration, right? Very efficient way to make ATP. But you do have another mechanism to produce ATP. What would that be? Not aerobic respiration, but
Fermentation?
yes, or anaerobic respiration. Perfect. Okay, let's think through why that might be. What's the function of a red blood cell again?
What was the question?
What's the function of a red blood cell?
To carry gases?
Exactly. So it's the function of a red blood cell is to carry the gases. And because it's carrying the gases, specifically oxygen, right? What does the mitochondria do? Mitochondria uses oxygen to produce ATP, right? So think of it this way. It's like putting me in a chocolate store. That wouldn't work. Why? Because I love chocolate and I would eat all of it, and that defeats the point, right? And that's why a red blood cell, because it functions in carrying oxygen, it can't consume it. And that's why it doesn't have mitochondria. Just like no one would hire me in a chocolate factory. Make
That
sense?
makes sense. Thank
Yeah,
you.
right? Okay. So it has no mitochondria. So as a matter of fact, it's the only cell in your body that undergoes anaerobic respiration at all times. Okay. It's also the only cell in your body that has no nucleus. So it's very unique. Okay. So does not undergo aerobic respiration. In other words, it doesn't use up the oxygen it carries. Okay. All right. That's a good start for structure of red blood cells and kind of understanding how the structure and the function are related to each other. All right. So now we're going to go through a life cycle of a red blood cell because that was one of the questions. All right. So we know that red blood cells are made up of millions of what? What are the functional unit of a red blood cell? Millions of them in a red blood cell. You said it. Whoever asked the question.
Sorry, what was the question again?
Sure. What's the functional unit of a red blood cell?
Heme?
Yeah,
Oh,
hemoglobin.
hemoglobin.
Okay. So the functional unit of a red blood cell is hemoglobin, because hemoglobins, are what transports gases. Okay. So let's take a look at the life cycle of a red blood cell, and then we're going to talk about how it breaks down. Okay. So red blood cell is composed of millions of hemoglobin. Okay. Now, where are red blood cells produced?
Bone marrow?
That's right. Okay, so a lot of you did your video on hypoxemia. Yeah. And hypoxemia is where you have low oxygen levels. So your body responds by releasing what from the liver and the kidneys. What's released from the liver and kidneys?
From the liver, you have bile cells.
Nope, in response to hypoxemia.
Oh.
Yeah. What's EPO short for?
Glycogen?
Not glycogen. EPO is short for erythropoietin. Erythropoietin. Okay, so when you have low oxygen levels, your body responds by trying to make more red blood cells. What stimulates red blood cell production is erythropoietin, and erythropoietin is produced by the liver and the kidneys. So whenever your body has low oxygen levels, and then in other words, it's undergoing hypoxemia, it's going to stimulate the liver and the kidneys to produce a chemical called erythropoietin. Erythropoietin is going to go to the bone marrow, and that's what stimulates red blood cell production. Okay, so erythropoietin stimulates the bone marrow to produce red blood cells. Now, the red blood cells are going to circulate through your body for about 120 days. And we talked about why. Okay, After 120 days, as it's squeezing through the blood vessels and going into the tissues, carrying the gases and so on, it's going to become damaged. When it's damaged, it's going to be collected in what organ?
The liver?
Yes, it can be in the liver, but mostly where?
In the lungs?
Not the lungs,
Gallbladder.
not the gallbladder.
Mm.
Keep going. It looks like this. And it's kind of like on the left side of your body.
So the pancreas?
Close. It's around that area.
Duodenum?
Nope, not part of your digestive.
Okay.
But good guess. Keep going. I mean, the duodenum is in that area.
I don't know. The stomach doesn't look like that.
Nope. You only have one of these, so it's not like you have a pair of them. The spleen.
Wow.
Okay, so the spleen is also referred to as the graveyard of red blood cells. Graveyard of red blood cells. And the nature of the spleen is that it has like these these fibers that form kind of like a filter mesh that catches the red blood cell and breaks it down. So after about 120 days, it's going to be caught by the liver or more likely the spleen where it breaks down. So when it breaks down, it's going to release all the hemoglobin. And the hemoglobin is going to break into its two parts. Oh, I think there is a question.
Yes, I have a question.
Yeah.
So what system is the spleen part of? So we've gone through respiratory, we've gone through digestive. I haven't studied the spleen yet.
Yeah,
So-
exactly. So that's a great question, Wendy. The spleen is part of your immune system. So we actually don't cover the immune system at all in anatomy and physiology, unless you take the second component of A&P, the advanced A&P. So that's why you should only have heard about it recently in the discussion of red blood cells, of where it's caught and it's destroyed. So if you
Okay.
check, yep. So this is the first time you've heard about the spleen.
All right,
Yeah.
thank you.
Yeah, absolutely. So the spleen is going to catch the red blood cell and it's going to help it to break down into its functional unit, which is the hemoglobin. Now, the hemoglobin has two main parts. It has the heme groups, and the heme groups looks like a pinwheel. And how many of them are there?
4.
Perfect. I usually don't make you know numbers, but when I do, you should know them. Okay, then what you'll find is that surrounding them is the globin chains.
Okay, so there's 4 heme groups and 4 protein chains called, and that's what the globin
Yep,
is, correct?
great. Yep.
Okay.
Okay. So there are 4 heme groups. And there's just a bunch of globin chains. Okay. Now the heme groups attached to the heme group. So the heme group essentially is made up of a pigment, and attached to that pigment is an element. What element is attached to the pigment that makes up the heme group?
Oxygen.
Not oxygen. Nope.
No?
Oh, but you're real close though, because this element has a high affinity and will bind to oxygen, which is why it's possible for red blood cells to carry oxygen.
Oh, I was under the impression that heme groups carry oxygen and the globin carries the carbon dioxide.
That's correct.
Okay.
Yep.
Is it iron?
Iron is it.
Okay.
Okay. So and that's why you have to have iron in red blood cells, okay, or specifically in the heme groups. So, and the reason for that is because iron binds to oxygen, which is what allows the heme group to carry the oxygen.
Mm.
Okay, so for each heme group, you must have one element of iron. Okay, so for each heme group, you must have one element of iron, which is why the heme groups carry the oxygen, where the globin chains carry the carbon dioxide. Okay. Now, when it gets to the spleen is going to release the hemoglobin, and it's going to break the hemoglobin into the heme group and the globin chains. The globin chains are just going to move on because the globin chains essentially are what? They're protein. So protein can be broken down to amino acids. And what's going to happen to them is that they're just going to be recycled. The amino acids are going to be reused. To form new proteins. Okay. Now, the heme group consists of the pigment and the iron. Okay. The iron is going to be recycled / reused unless it's lost in what?
In the kidney?
No, not necessarily. I'll give you a clue. It's lost in a particular gender during a certain time of the month.
Oh, no.
Menstruation?
Yeah. And that's why women tend to get more anemic, right? Because they lose blood during menstruation. And during that menstruation process, they also lose the iron.
Oh.
Yep. So the iron should be recycled and reused to make more heme groups, to make more hemoglobin, to make more red blood cells. But oftentimes it's lost during trauma or menstruation. Okay. So if you have like bleeding, if you have trauma where you have a cut, you can also become anemic that way. Okay. So that's what happens to the iron. It's either recycled or reused or lost during blood loss. Like menstruation, trauma, which then makes that person anemic. Okay. The pigment, however, goes to the liver. The pigment goes to the liver and gets kind of converted to bilirubin, bilirubin, and then finally to bile. Okay, so the pigment goes to the liver and gets converted to bilirubin and then finally to bile. And keep in mind that bile is going to then connect with the digestive system that we'll talk about tonight. Okay, so that's what happens when the heme group breaks down. So the heme group breaks down. It's got the pigment, which goes to the liver, and then the iron, which is recycled, where the globin chains break down to the amino acids, and that's just reused to make more protein. Okay, so that's the life cycle of a red blood cell. It's produced in the bone marrow, and how that happens is that it's stimulated by erythropoietin, which is released by the liver or the kidneys. Once it's produced, it's going to circulate through the body for about 120 days. It's going to be caught by the liver, or most likely by the spleen, and then it's going to be broken down into the hemoglobin, and then the hemoglobin is going to be broken down into the heme group, the globin chains, and then they have different fates depending what they are. Okay? Questions about the life cycle?
Thank you. That really helped.
Yeah. Okay. All right. So make sure, so that's just the erythrocytes, okay? Make sure that you go through what plasma is, the function of plasma, the components of plasma, and the plasma proteins, okay? Within the plasma proteins, you should know that there are three. There's actually more, but I only require you to know three. One being fibrinogen, two being albumin, three being globulins, and there is a mistake in the lecture, the script. The person who did the script said that the globulin functions all the same, but they don't, because you do have three types. You have the alpha and the beta, and the alpha and beta both function in transportation, where the gamma functions in immunity. Okay, so that is one correction in the script. Okay, so within the globulin, there's alpha and beta. That's the ones that do transportation, where the gamma is the one that does immunity, because they are forms of antibodies. Okay, now anytime you hear the term ogen right here, and you're going to hear it pretty often, right? You heard it in the digestive system as well. So the term ogen refers to inactive. Anything that has an ogen in it means it's currently inactive. So it requires a chemical to convert it to, so it's going to go from fibrinogen, then it's going to become fibrin, and that's when that protein is going to cause clotting, which makes absolute sense, because you don't normally want to be clotting, right? That could potentially kill you. And that's why clotting is a positive feedback, not a negative feedback, okay? So originally, it's in the form of fibrinogen. It's a plasma protein in blood in the form of fibrinogen. And if you have a cut in your blood vessel, there's a protein on the outside that goes in that converts the fibrinogen into fibrin. And that's when it should clot and hopefully seal out that nick or that cut in your blood vessel. Okay, so that's what fibrinogen does.
So that's
Yeah, go ahead.
sorry. So that's why fibrinogen is inactive, like initially, and then it has to go through everything.
That's right.
Okay.
Yep. Great job, Victoria. Yep. Now the albumin is responsible for viscosity and osmolarity, because it's a plasma protein that controls how much water stays in the blood or how much water leaves the blood. Okay. A practical use of albumin. Like my dad, he used to receive paracentesis. So they would suck the water out of his abdomen, because his abdomen would, retain water, right? So they would take out all the water, and what would happen then is that it would cause his blood vessels to draw more water out, which then lowered his blood pressure. And we didn't want his blood pressure to get too low, right? So they would give him albumin. And when they gave him albumin, it helps to keep the water in the blood vessel and hence keep his blood pressure at a normal level. Okay, so that's why albumin influences osmolarity, in other words, how much water is retained in blood, and the viscosity, how thick the blood is. Okay, so albumin is something that you give to patients that undergo paracentesis when you take out too much water from their bodies. And that's because you want to make sure that there's enough water in their blood vessel, because you know that water influences blood pressure. Okay, all right. So those are the three plasma proteins. Make sure you know leukocytes, white blood cells. I have the mnemonic that I always use is never let monkeys eat bananas. It's really great because it tells me what they are. N for neutrophils, L for lymphocytes, M for monocytes, E for eosinophils, and B for basophils. It also tells me the order of most abundant to least abundant. So of these white blood cells, the most abundant one in your body in blood is neutrophils. The least abundant is basophils. Okay. Another mnemonic to know is Beng, B-E-N-G. That's because white blood cells are categorized into granulocytes and agranulocytes. If they're granulocytes, they have these protein granules on the inside of their cells, so it looks like it's dotted. So the ones that are granulocytes include basophils, eosinophils, and neutrophils, which means what are agranulocytes?
Can you repeat the second? Whatever you call a mnemonic.
Sure, Beng, B-E-N-N-G, Beng, like B-I-N-G, it's
okay.
Beng. Yeah.
Thank you.
No. So what's not B, what's not E, what's not N?
The agranulocytes.
Yep, which are what?
Those do not have the cytoplasmic granules.
Yep, which leukocyte would those be?
The last two remaining.
Which are?
It's like dragging it out.
That's my job. It's okay. Which are?
Lymphocytes and monocytes.
That's it. Lymphocytes and monocytes. And that is a test question. So you do need to know what's granulocytes and which are agranulocytes. Okay? All right. We don't have time for it because I still have to go through respiratory. So make sure you know what they are. For example, if you had a bacterial infection, which one of these would be elevated?
The neutrophils.
Exactly. Neutrophils function in fighting off bacteria. Okay. Yep. Perfect. And there's normally, if we had more time, I'd tell you stories about each one, but there's a really cool one, and that has to do with eosinophils. Okay. So anytime you have elevated eosinophils, it's tragic because it means you've got some type of parasite. So if you ever have a chance, watch Monsters Inside. Okay. So there's a show called Monsters Inside where there was a case where this person had really bad headache after swimming, and it turned out that they had this water parasite that goes through the brain and was causing meningitis. And people had thought it was bacterial, right? So they gave them antibiotics, but it didn't help. And it turned out it was parasites. And they didn't find that out until they did the blood draw, and they found it was eosinophils that were elevated, which means it was a parasite, not a bacteria in the brain of that person. Cool show. Monsters Inside of Me. Okay, moving on. Lots of cool stories. But anyways, the heart. So moving on to the heart. For the heart, make sure you know the structure, the function, and the blood flow. And oftentimes when I go through the heart, I do all three at the same time. Okay? So doing the structure, function, and blood flow all at the same time. Okay? So I only have about 10 minutes left, so I'm going to move straight to respiratory. But before I do that, make sure you also know about cardiac conduction and EKG. A lot of you did a great job on that, so watch each other's videos. Blood vessels use this cognitive map to kind of differentiate between arteries, veins, and capillaries. And then with this one, the presentations on these did a great job at giving an overall kind of synopsis of the difference comparison between arteries, veins, and capillaries. But a lot of the people who did this chose this topic for the videos, didn't really go into the layers really in detail. So make sure you know that you are responsible for the layers. So make sure you know the layers, okay? How they differ between each one of those. I did a cognitive map for you. I would recommend that you like delete it, go through it, and try to do it on your own, and then compare it to the answer, okay? All right. On to respiratory. All right. So within respiratory function, make sure you know the functions and structure of the respiratory system. That is really difficult initially, and that's why your worksheet on the respiratory system had all of that, had that long table. Okay, so you know like the parts of the respiratory system, the accessory structures, as well as the tissue lining. Okay, now you shouldn't have to memorize them. It should make sense. Okay, so let's go through an example of one. I don't have it here, but let's, that tells me I need to go into my next meeting, but let's quickly go through this. So, for example, if you did, let's go with what are the parts of the respiratory system? So you start with the nose. What occurs after the nose?
It warms up.
So that's the function. So first we're gonna go through the organs first. So a good mnemonic is please let's talk. So what's next after the nose?
Is it the larynx? Larynx?
Nope. Please let's talk.
Pharynx.
There you go. The pharynx, and then the larynx,
And the trachea.
and then the trachea. That's great. Okay. So make sure you know what they do, the function of each of these structures, the accessory structures, as well as the tissue lining. Okay. Now, with the tissue lining, it's actually pretty straightforward, and it makes sense more than memorization, because here's the thing. Anytime you're looking at the upper respiratory tract, okay, 95% of the time, the upper respiratory tract, if it's a pathway for air only, it's going to be lined with what?
Is that the pseudostratified columnar epithelium?
Yes, ma'am. Pseudostratified columnar epithelium. 95% of the time, if that structure only allows for air to go through and you choose pseudostratified columnar epithelium, you are correct. Okay.
Is that because
Yeah, go ahead.
the cilia and then you got the goblet cells?
That's right.
Help with all that? Okay.
That's exactly why. It makes sense because pseudostratified columnar epithelium is associated with cilia, which helps to, these hair-like structures that help to move things along, and the goblet cells that produce mucus. Okay. And that's why your upper respiratory tract is oftentimes lined with this tissue, because it has the cilia and the mucus. That's right. Okay. Now, if it allows for other things like food and liquids, then it's going to be lined with what?
The simple stratified squamous.
Definitely not simple, because it's going to have abrasion, because the fluid that moves through can scratch the lining. So it's got to be not simple, but
Then the stratified.
that's right.
Squamous. Yeah. Okay. And that's because it's at the mouth, so it needs to be harder, right?
That's right. Yep. So you can see it makes sense. It's not like, oh my God, the pharynx is lined with what? The larynx is lined with what? Just ask yourself, does air only move through it, or does food and air move through it? If your answer is air only, it's pseudostratified. If your answer is food or liquids, and because of the abrasion, it's oftentimes stratified squamous. Okay? All right. Then ventilation and gas exchange. I only have time to go through one. So which one were you struggling with?
Ventilation.
Ventilation, excellent. That's one I would choose as well. Now, I know I'm going through this kind of quickly, but I will say that this is exactly the point where I had to go online for one of my in-class sessions during COVID. So I have a recording of all of these things. So if I skipped over anything, you can watch my in-class lecture that I recorded online. Those lecture recordings are on the announcements, so make sure you watch them. Have anyone had a chance to look at them yet?
Yes.
Perfect. Okay. So
Very helpful.
excellent. I'm so glad you found them. Okay. So because I would normally spend about like 3 hours on each of these chapters that I'm only have like 15-20 minutes on, and that's why it feels a little incomplete, and that's why I highly recommend that you go and watch those videos. Okay. All right. So let's do ventilation. All right. So in order to understand ventilation, you need to know a couple of things. Okay, so there are some rules you need to know first. The first rule that you need to know is called Boyle's Law. What's Boyle's Law?
Has to do with volume and pressure
Yes, ma'am?
connected somehow.
Yes. So Boyle's Law say that volume and pressure are inversely proportional. Which means when one goes up, the other one goes down. When one goes down, the other one goes up. Makes perfect sense. Okay, let me give you a demonstration of that. When you take a can of soda, or if you're from the north, you would call that pop. So a can of soda or a can of pop, okay? If you shake it up, what happens when you open the can?
Explodes in your face.
It's going in your face, exactly, because the fluid that's in that can, okay, because the fluid in that can fills up the entire can, okay? So because the volume is low, the pressure is high.
Mm-hmm.
If I took that exact same fluid and instead of that can, I put into a gallon of milk, it's the same amount of fluid. I can shake that gallon up, but would it explode in your face?
No.
No. Why? What did I just do?
You increase the volume, so decrease the pressure.
That's exactly it. Make sense?
Yes.
Another way of looking at it is this. It's about particles bumping into each other. So if I have a room that's only this big with 3 particles, the likelihood of those particles bumping into each other and creating the pressure or the force is a lot higher than if I put those 3 molecules into a size much larger. So then the chance of those molecules bumping into each other is a lot less.
Mm-hmm.
Okay, because there's more room to move. Okay, so pressure is essentially force. Okay, so when two particles bump into each other, that's what creates force and pressure. Okay, but if the volume is big, then the likelihood of them bumping into each other is a lot less. But the volume is smaller, then they're more likely to run into each other. Okay, so that's why Boyle's law of volume and pressure being inversely proportional makes complete sense. Right?
Yes.
Yeah.
Okay. The other rule that you need to know is diffusion, and this one you already do know. What do you know about diffusion?
Goes from higher to lower.
Yes, ma'am. High to low, always high to low. Okay? So that you already know. All right, so now let's apply it to, one more thing. So you know Boyle's law, diffusion, Now you also need to know about the muscles involved in ventilation. What are the muscles involved in ventilation?
Diaphragm and intercostal muscles.
And intercostal muscles. Perfect.
So the main muscle will be the diaphragm here, not the intercostal muscles.
They both are, but it's the diaphragm that you can directly influence.
Because I got that question wrong in the quiz, and it was diaphragm, the correct answer.
Yep.
Okay.
No, it's true. So they're both important, but the only one that you can influence is the diaphragm.
That's why they call it the main one. Okay.
Yep, that's right.
Okay.
Okay. So here is this person right here. Okay. And here's your diaphragm that's neutral. When the diaphragm is neutral, it's slightly curved. Okay. All right. So you have atmosphere pressure, and atmospheric pressure does not change. Okay. And what's atmospheric pressure?
Is it 760, I don't remember, millibars or something?
Millimercury. Yep. Very good. Now, and then you have the lung pressure. Okay. Now, when the diaphragm is in its neutral state, the lung pressure is also 760 millimercury. Okay. So if the diaphragm is if the diaphragm is in its neutral state, nothing happens. Why? Because the pressure of the atmosphere in the lung is exactly the same, right? So diffusion doesn't happen because it's equal.
Yeah.
It's an equilibrium. So what we do is that we can either contract the diaphragm, and when we contract the diaphragm, we make it shorter. So the diaphragm, when it contracts, it gets shorter, it pulls downward. Okay. When we relax the diaphragm, it gets longer, and when it gets longer, it goes up like this. So that's the difference. So this is when the diaphragm relaxes, and this is when the diaphragm contracts. Okay, so let's pick one. So we know what happens when the diaphragm is neutral. When it's neutral, nothing happens because the pressure is the same. But let's say what happens if the diaphragm contracts. So when the diaphragm contracts, what does it do to the volume? Did the volume go down or did the volume go up?
Increases.
Yeah, volume increases. And when the volume increases, Boyle's law say that what happens?
Pressure decreases.
Yes. And as a matter of fact, now the lung pressure is going to decrease to 756 millimercury. So now after the diaphragm contracts, if you compare the pressure of the lung to the pressure of the atmosphere, which is greater?
The atmosphere.
So what's going to happen in diffusion?
The air rushes into the lungs.
That's right. Okay. A lot of people think that when I go like this, that the air goes in and the air causes my lungs to expand, but it's not. It's actually the opposite reverse. It's because my diaphragm contracts that causes a vacuum that pulls the air in. Okay, so the misconception is that when you breathe in, the air goes in and that's what causes your lung to expand. That's not correct. Now we know, right? We know how you inhale. You inhale because the diaphragm contracts, which changes the volume and the pressure, which causes the lung pressure to be below the atmospheric pressure and creates a vacuum that then pulls the air in. Yes, no?
Yes.
How you feeling about that?
So basically, when the abdomen contracts, what happens to the intercostal muscles? They move up or to the sides, right?
Yes.
They move to the sides, so that makes it go bigger, and that's why the air comes in,
Yeah. The pressure in the lung decreases. And then
decreases.
when the pressure of the lung to the pressure of the atmosphere is compared, the pressure of the atmosphere is higher. So in diffusion, things go from high to low. So then it goes in.
Okay.
Yep.
So
Okay.
yeah, because the volume increases, the pressure decreases, and then comes in, right?
Yes, ma'am. That's
Okay.
right. So now let's look at what happens in relaxation. So in relaxation, what does it do to the volume?
Decreases,
Yeah. And the volume of the lung decreases, what happens to the pressure?
increases.
So as a matter of fact, the lung pressure moves from 760 to 763 millimercury. So now which is greater?
The lung pressure and yeah, the lung pressure.
Yeah. So then the air is forced out.
Okay.
And that's
Do
what
we
we
need to know the difference of 763, 756?
you do need to know those numbers, yes.
Okay.
Yep. So we were able to go through the respiratory system and cover the ventilation. I'm going to spend a little time talking about the gas exchange as well and go into the digestive system. And then hopefully we'll still have time at the end and not go over to do the practice quizzes. Does that plan sound okay with everyone?
Sounds good.
All right. So in gas exchange, we have two types. What are the two types of gas exchange that occurs?
Oxygen and carbon dioxide?
Yes, it is gas exchange between those two. I guess what I'm asking more of what location or where does gas exchange occur? What two main locations?
Oh, the capillaries and the alveolars?
Yep. And then where within the body?
That's in the lungs?
Yep. So we have pulmonary.
Alveoli?
Yep. So pulmonary gas exchange. And we have what else? Where what's that term that we use where it occurs the rest of the body? So pulmonary gas exchange occurs between at the heart, so between the lungs and the blood vessels. So what's the other one called?
Is it systemic?
There you go. Systemic gas exchange. Okay, so two forms of gas exchange: pulmonary gas exchange and systemic gas exchange. So pulmonary gas exchange between the lungs and blood vessels. Where systemic gas exchange occurs between the body and blood vessels. Okay, so let's start with the pulmonary first. I'm going to make a line to divide the difference. Okay. So here we're looking at something like this. Start with blue. Mm, and then something like this. And then and when we talk about the lungs, someone has said the alveoli, and that's exactly correct. So here are the alveoli. Okay. All right. So in the lungs, you have the alveoli, and that's where gas exchange occurs. And remind me, what are the cells of the alveoli again? The alveoli consists of what cells? There's 3 cells that make up the alveolar lining. Oh, am I still online?
Yes. We're thinking.
Okay, sometimes I get cut off and I'm still talking for the longest time and there's no response, like, oh wait, and I got cut off. So
Is it squamous alveolar,
yes,
and then
it
great
is.
alveolar, and then alveolar macrophages?
Excellent. You got it. Those are the 3. Thank you. So you've got simple squamous or squamosal alveolar cells. Alveolar cells. You have the great alveolar cells. And then you have the macrophage alveolar cells.
The thing that confused me with this is they call them like type 1 and type 2, and they have multiple names. That always kind of confuses me.
That's right. I agree. So the type 1 is the squamous. The type 2 is the great alveolar. And then the macrophage alveolar cells used to be called the dust cells. So many names, right? So of these 3, the squamous alveolar cells, that's the one that allows for gas exchange. That's why it has to be nice and thin, simple squamous. The great alveolar cells play an important role in producing surfactant. And that's so that when the alveoli, when you exhale and the air leaves the alveoli collapse. And when they collapse, you don't want them to stick together so that they can't expand again. So you have to have the surfactant there. And that's a big problem in newborns because newborns, their lungs aren't functional quite yet, so they haven't produced the surfactant. So with newborns, in the past, their mortality rate was, very high. Is very, when you had a newborn, they were very likely to die because at that point in the past, we hadn't made any artificial surfactant yet. So they couldn't breathe when they were born because their lungs wasn't functional yet. Luckily, through medical inventions, we've made the surfactant. So now newborns, not newborns, but preemies, premature babies, have a much better chance of survival because then we just, squirt in a spray of the surfactant, and that allows their alveoli to expand again until they can start to produce their own. So that's the importance of the great alveolar cells. Surfactant is really important. That prevents the alveoli from sticking together and so that it can expand again. Okay, so surfactant. And then the macrophage alveolar cells, those are very important as well because, we're going to breathe in air that's contaminated, so to some extent your alveoli can protect you because the macrophage cells are like immune cells that are permanently there. So what they do is that if they detect anything that shouldn't be there, they're called macrophage because they eat them. They eat things that shouldn't be in there. Now, there are certain conditions that have microorganisms that can survive inside a macrophage. And those, a really popular example of that would be tuberculosis. So tuberculosis has a bacteria that can stay, even though you inhale it in and it makes its way down to your alveoli. Macrophage digests it, but that bacteria is able to survive inside your macrophage, where other things get broken down. Okay, so it's able to survive there until like you get older or you're immune suppressed, and then they break out of the macrophage and then they cause damage to the lungs. Okay, so your alveolar macrophages are really important because they're your immune cells that are permanently there, because over time your body has developed to a point where it knows that the air that you breathe in can be, contaminated. Okay, so that's the role of these three cells. But the one that we're really interested in is the squamous alveolar cells right now, because that's the one that allows for gas exchange. Okay. All
So
right.
for test purposes, should we know it by squamous alveolar or type 1?
Unfortunately, you need to know it by both.
Oh, okay.
Yeah, I know. I know. All right. Then here you can see that that's why the heart is right next to the lungs, right? Because the heart is going to pump blood to the lungs. And when it pumps blood to the lungs, that blood is moving away from the heart. So what kind of blood vessel is that? Oh, hold on, let me get some. Yes,
Artery.
Exactly. Arteries always move blood away from the heart. Okay. So arteries always move blood away from the heart. So what would we call this artery right here? If it's an artery, it's moving from the heart to the lungs, it's always going to be called what?
Arteriole?
No. It's the pulmonary artery.
Oh, I thought you were talking about it in the
The
capillaries.
Yeah, not yet. I will soon though.
Okay.
So the pulmonary artery. Okay, so the pulmonary artery is going to take the blood from the heart to the lungs. And then what blood vessel takes the blood from the lungs back to the heart?
The pulmonary veins.
Perfect. Pulmonary vein. And then in between, let's do purple, I don't know what about it. We have the capillaries. Because the capillaries contain the simple squamous cells that allow for the gas exchange to occur. Right, so between the capillaries and the squamous alveolar cells, that's what creates the respiratory membrane. Okay, so the respiratory membrane is between the capillaries and the squamous alveolar cells. And then there's a membrane in between them. Okay. A basement membrane, to be specific. Let me change that. Oops. So this right here is what forms the respiratory membrane. For exchange. Okay. All right. So the heart's going to pump blood from the heart to the lungs through the pulmonary artery. So the pulmonary artery is going to be high or low in oxygen. The pulmonary artery is going to be high or low in oxygen.
High.
What? If it's high in
High.
oxygen, would it need to go to the lungs?
No, then it's the opposite. Yeah, you're right.
There you go. So it's going to be low in oxygen and high in carbon dioxide. And that's why it goes to the lungs. Okay? Because whenever you breathe in, even though you learn that the composition of air is highest in what? What's the highest component in air?
Nitrogen?
Yeah, exactly. So oxygen isn't even the highest, but it's higher than what's in your body. Okay? So
So
when
we can breathe nitrogen and nothing happens to us? It just comes in and out?
it's the composition of air overall. It's not pure nitrogen.
Okay.
So it's partial pressures, right? We're not talking about atmospheric pressure. We're talking about partial pressures. Okay. But yeah, so the composition of air. So when you breathe in air, it does have other gases. Okay. But it's still relatively higher in oxygen than carbon dioxide than in our bodies. So the air that you breathe in is going to be higher in oxygen and lower in carbon dioxide. And the reason for that, by the way, just because I'm an environmental scientist as well, is because even though we produce carbon dioxide and that's what we breathe out, there's a check and balance in our environment that uses the carbon dioxide and produces oxygen. What are those things? Beautiful things called.
Plants and trees.
Yes, plants, trees, algae, they all take the carbon dioxide and they use that for their metabolism and then they produce oxygen. So go and plant a tree. Okay. All right. So when you breathe in, the composition of air is higher in oxygen compared to the carbon dioxide than it is in your body. Okay? Because we use oxygen and produce carbon dioxide. So that's why the pulmonary artery is going to bring in blood that's low in oxygen, high in carbon dioxide. And when it becomes a capillary right here, in this area right here, there's going to be an exchange. So the oxygen in the lungs, the oxygen in the lungs is going to move into the blood vessels, the capillaries, and the carbon dioxide right here is going to move out into the alveoli, and that's what you exhale. So you exhale higher carbon dioxide levels than before, than when you inhaled. And the oxygen that you inhaled is going to transfer then into the capillaries. So that's how gas exchange happens in the pulmonary gas exchange. Questions? So the blood that returns to your heart through the pulmonary veins now is going to be high in what?
Oxygen.
Yes, ma'am. And low in carbon dioxide. Does that make sense? Questions? Questions? We're good?
We're good.
Okay, let's move on. Then in the body, then the reverse happens, right? Because in the body you have these cells, the cells of the body. The cells in the body is actually going to use up the oxygen in metabolism and produce high levels of carbon dioxide. So here it's going to have high levels of carbon dioxide and low levels of oxygen because it's used it. So the blood vessels that move through the body then initially when it gets to the parts of the body, it's going to be high in oxygen and low in carbon dioxide. Okay, so the arteries that go into the body, you have the renal artery, you have the hepatic artery, all the different arteries that enter into the different areas of your body. It's going to start out being high in oxygen and low in carbon dioxide until it transitions from an artery to a what? To a capillary. And then when it leaves that area, it leaves as a what? To go back to the heart. Oops, not pulmonary. As a
It leaves
vein.
as a vein, no?
That's right. You are right. Okay. So when it leaves now, and gas exchange, the systemic gas exchange that occurs is going to have it pick up carbon dioxide and drop off oxygen. And thus, when it leaves, it's now high in carbon dioxide and low in oxygen. That's why it has to go back to the heart so that the heart can then circulate it to the lungs and have the pulmonary gas exchange occur. Does that make sense? Questions? So when you talk about gas exchange, you have to ask, are you talking about pulmonary gas exchange or systemic gas exchange? Because different things happen in the different gas exchanges. It still involves blood vessels and then either cells of the body or cells of the lungs. Okay. Questions?
And this all occurs through diffusion.
Yes, because it's high. It's going from high to low. That's a great question. Thank you. Yep, and that's why you notice that it's high to low. Mm-hmm.
Is there a respiratory membrane for the systematic gas exchange?
There is, yes.
So
Yeah.
that will be the capillaries and the arteries?
No, it'd be the membrane that allows for the gas exchange would be between the capillaries and the cells of the body.
The cells of the body. So that basically just depends where you're at, right?
That's why we're not being specific about it. Exactly.
Okay.
Yep.
Thank you.
Yep. Where with the pulmonary side, we can be a lot more specific. Yep. Okay. All right. So that concludes the respiratory chapter. So in the respiratory chapter, make sure you know function, structure of the respiratory tract. Doing that flow chart, it would be very useful. We talked about ventilation this morning or this afternoon, and then we finished it with the gas exchange recently. Okay. So with that, I'm going to move on to the digestive system unless anyone has questions. All right, let's move on. So in the digestive system, make sure you know the functions of the digestive system. Just to remind me, what are they again?
Ingestion, digestion, absorption, compaction, and defecation.
Perfect. We'll start with ingestion. That's one of my joys in life, right? Eating. By the way, digestive system is my favorite chapter because it's just so fun. So ingestion is eating. And when you ingest your food, it can occur through what process? You have to ingest it and then you have to chew it. And so what's that chewing called?
Mechanical.
breakdown.
Yeah. So mastication. So that's the chewing. And that's why in your mouth you have the masseter muscle to help you chew. So kind of helping you remember these terms. Then after ingestion, you have digestion. Oops, Yes, I was going to ask you instead of giving you the answer, but okay. So you have digestion. So that's the breakdown of the foods that you've ingested. And it happens through two mechanisms. And what are they again? You kind of talked about it a little bit earlier.
Chemical and mechanical breakdown.
Perfect. Chemical and mechanical. So whenever I ask you about digestion, you have to tell me which form. It could be both. It could be one or the other, or it could be both. Okay, so for example, in your mouth, what digestion happens?
Both.
Both. Exactly. Mechanical through the mastication, the chewing and grinding of your teeth, and chemical because why?
Saliva.
Yes, and specifically what in saliva?
So like the acids?
Not the acids.
Amylase.
Yes. So if there's ever chemical, you have to tell me the specific chemical that's doing the breakdown, because different chemicals break down different things. So mechanical breakdown is going to break down any kind of food that you put into your mouth. Where chemical digestion, only one thing happens in the mouth, right? Because so in chemical, if we're looking at the mouth produces the saliva and specifically contains salivary amylase, which only breaks down what?
Carbs. Delicious
That's
carbs.
right. Exactly. So of all the foods that you eat, carbohydrates are the first to be broken down chemically. Which makes sense why if you eat carbs, you feel hungry faster because it's broken down a lot faster.
Mm.
Where protein, like meats, don't get broken down until it reaches your stomach. So they exist in their original form a lot longer, so you feel full longer. Okay? All right. So whenever I ask you for chemical, you have to be specific. What chemical and what specifically is being broken down. All right? So that's 2. And then what happens after digestion?
Absorption.
Yes. This concept is kind of difficult because a lot of us think that when we eat, we brought it into our bodies, right? No, because your digestive tract or your alimentary canal is one tube that goes through your body, but it's not in your body until you have absorbed it. Okay, so from the mouth to the anus, if you follow it, it's one closed tube. The tube from one end to the other end. Okay, so in order for the food to really be in your body, you have to absorb it, which means it's got to be transferred from your digestive tract, that tube that goes through your body, but that is not in your body. Okay, so the tube that goes through your body, so the food has to move from that tube into the blood vessels, and then once it gets into the blood vessels, then it can be circulated throughout your whole body, and that's when it's in your body. So that's what absorption is. You gotta move the food that you've eaten, okay, to your body. Okay. So absorption is when it's referred to as the uptake of food, of nutrients into body. Okay. And then once that's done, whatever remains, what happens to whatever remains?
Compaction.
Yep. And I actually like to combine the two, compaction and defecation together. Because that's kind of like the final step. So everything gets squeezed into like small components, and then it gets pooped out, defecated. Okay. All right. So those are the functions of the digestive system. Make sure you go through and list the digestive tract. That's that long alimentary canal from the mouth to the anus. Make sure you know the layers and the linings of those layers. And that's that flow chart that I had you do in the worksheet, okay? So just remind me, what are the layers again? Let's go from deep to superficial.
Mucus,
Yes, mucosa,
mucosa, submucosa,
Muscularis and serosa.
perfect. I am so thrilled. Yes, you did a great job. Exactly. Okay, so do the exact same thing on the test. Okay, so mucosa to submucosa to muscularis to the serosa. Okay, then know what lines those layers as well, and kind of know the distinction between them. Now these linings right here. What's really interesting is because it's the same tube that goes from the mouth to the anus, but the linings change as you get to the different organs, and that's why the organs are called the way they are. Is because there's differences in them. So, for example, you'll find the rugae or the rugae, plural or single, okay, only in the lining of which organ?
stomach.
Exactly. Okay, so that's part of the muscularis layer. In addition to that, and that's why the stomach lining, the muscularis lining of your stomach has not just 2 muscle linings, but actually 3. What are those 3 again?
Longitudinal, circular, and oblique.
Perfect. Exactly. So that's the distinctions that you have to make within these linings as you move from one organ to the next of the alimentary canal. Okay, you did great. All right. So that's what this next one is kind of walking you through. So we start with the mouth. What is it lined with? Describe the following structures: the tongue, the lingual papilla, the lingual frenulum, the palate, the uvula. We had a fun conversation about the uvula this morning, right? And then describe the structure and layers of the teeth, the crown, the root. What are the 3 types of salivary glands, and what do they secrete? Okay. Then you move on to the pharynx. Not much about the pharynx, right? Because it's just a pathway. Then you move to the esophagus. Understand what peristalsis is, what esophageal sphincter is, and then we move on to the stomach. And that's when it starts to get really serious, the stomach. Okay, so the stomach is one of the key players, and that's why we get a lot more detail. What are the regions, like the cardiac, the fundic, the body, the pyloric? What are the gastric pits? What are the cells that line the stomach, like the chief cells, the parietal cells, and so on? And what do they do? How is food digest? How is the digestive tract of the stomach different? And we just talked about that, right? It has the rugae. It has the 3 instead of 2 muscle lining. It has 3. Describe the accessory organs. What's the pancreas role? What's the liver's role? What's the gallbladder role? And know how they connect. And that's through the ducts, right? You've got the cystic duct. You've got the common bile duct. Describe the function of the small intestine. The small intestine is massive. So much things happen there. So that's another major one that you have to know. Okay. What are the circular folds? What's the villi versus what's the intestinal crypts? Whoever named this was very, into sci-fi. Look at gastric pits, intestinal crypts. Describe the function of the large intestine. What's the segments? What's the role of microbes here? Okay. And make sure you know how chemicals are digested chemically. And usually students have the most difficulties with that. So that's the one I'm going to spend the rest of the time on, unless there's requests for other things as well.
I just have a quick question.
Yeah.
Is it safe to assume that our feces is fermented?
feces is fermented. No, I wouldn't use the two terms together because fermentation refers to a process where oxygen is not present.
Okay. So
Yep.
the bacteria is not fermenting the feces at all. It's just basically just eating anything that is not that wasn't
So
absorbed before.
yeah, so there is one component in the foods that we eat that we can't break down, and that's cellulose. So we don't have the enzymes to break down cellulose. We can break down starches, but we can't break down cellulose. That's plant cell wall, and that's why when you eat like a kernel of corn and you didn't chew it, guess what you see when it comes out the other end? The exact same kernel of corn, right?
Yeah.
I've noticed it. Be honest, I'm sure others have noticed it too. Yeah,
No, definitely. Yeah.
yeah. And that's because our bodies don't have the enzyme to break down cellulose, but the bacteria in our gut does. So the bacteria in our gut break down the cellulose and they use it for their nutrient. And the byproduct of that is a chemical we cannot produce ourselves. Does anyone know what that is? What does the bacteria produce for us? And that's why you still have to have absorption occur in the large intestine.
So when you say gut, are you referring to the large intestine?
Yes.
Okay.
Vitamin K.
Oh.
So that's in your notes to make sure you kind of review that again. So we can't make our own vitamin K. So we rely on the bacteria in our large intestine to break down the cellulose and release vitamin K. And then we absorb that vitamin in our gut, in our large intestine. And then we need that. Okay.
All right.
All
Thank
right.
you.
Yeah. So lots of cool things. But that question about fermentation is interesting. It tells me that you know a lot about microbes because you're right. Microorganisms tend to, use up the things and hence cause a little bit of gas to occur. So a little bit of fermentation happening there. But our bodies do not ferment the feces. It just
Okay.
compacts what's there and then releases it.
No,
Yeah.
that's exactly why I was thinking it was fermented. But okay, thanks for clarification.
Yep. Okay, great question. Great connection, though. Okay. All right. Other questions before we move on to the chemical digestion? All right. If you do, ask anytime. This is your session, so it's open. Let me make that a little bit clearer. I'm going to show you this other thing. Here's digestion. Here we go. All right. So in chemical digestion, I had you use my favorite candy, which is Almond Joy, because Almond Joy has the three main ingredients that your body digests. It's got the chocolate, which is carbohydrates. It's got the almond, which represents protein. And it's got the coconut, which represents lipids. So it's a really good way and a fun way to figure out how things are broken down. Okay, so let's do that together. Let me make this, change the size of this a little bit. So that I can change the size. All right. So starting with the mouth, you have the tongue, the teeth, the salivary glands. So in the mouth, you have ingestion, which is the eating, the best part, reason for living. Okay. And then you have digestion. So you do have mechanical digestion happening. That's the chewing, the mastication with the teeth. You also have chemical digestion occurring through the saliva, specifically the salivary amylase. Now you'll notice as we go through this, anytime you see something that ends in an ACE, oh, where's my pen? Oh, darn it, my pen's not working. it's not working. So another charge for it maybe. All right. So anytime you have oh, there we go. It's working now. Anytime you have this ace right here, if it ends in an ace, it's telling you that it's an enzyme. If it ends in an ose, it's telling you it's a carbohydrate. Just like earlier this morning, we talked about if it ends in an ogen, it tells you it's in the inactive form. Okay? So pay attention to the words because sometimes it gives you clues as to what it is. So salivary amylase, amyla meaning carbohydrate, is an enzyme that breaks down carbohydrates. So in other words, the chocolate at this point is what's broken down first chemically in the mouth. Okay? Then we have, it goes through the pharynx, specifically the oral pharynx and the laryngeal pharynx. It doesn't go through the nasal pharynx, right? Because that's for air only. So it's a pathway. And then it goes to the esophagus. It's also a pathway, but you should know about the peristalsis that happens here. And that's a smooth muscle contraction that helps to kind of move the food down. Smooth muscle contractions. Okay. And then it reaches the stomach. Okay, we're going to spend some time with the stomach. Okay, so what happens in the stomach? What function happens in the stomach?
Chemical
Yeah.
breakdown?
So you have digestion. Okay. You have digestion occurring here. Okay, and you also have what kind of digestion happens here? Is it just
Both.
chemical or is it mechanical as well?
Mechanical as well.
Yeah. So mechanical digestion occurs through the churning of the stomach rugae. And then you have chemical digestion happening. And remember, whenever you do chemical, you gotta tell me the actual chemicals. So what are the actual chemicals? Oops.
Pepsin?
It actually starts off as what first?
Pepsinogen or pepsinogen?
Yep. Pepsinogen.
Pepsinogen, okay.
And remember, if it's an ogen, that means it's currently in its,
Inactive.
that's right. And in order to activate it, what do you need?
Hydrochloric acid.
That's right. So hydrochloric acid activates it, and then that's when it becomes pepsin. And pepsin does what?
Breaks down protein.
Yes. And what does it break the protein down into?
From polypeptides to polypeptides,
Yeah.
right?
Perfect. So not amino acids quite yet. Okay, so it does break it down to the peptides and the polypeptides. Great. So that's going to break down what part of my favorite candy.
The almond.
That's right. All right. Okay. Then you also have hydrochloric acid. In this case, the hydrochloric acid is used to convert the pepsinogen, but it is also used to create an acidic environment in the stomach. To do what? What's your food always going to have, no matter how well it was prepared?
Bacteria.
Yes, ma'am. Yep. So the hydrochloric acid helps you control the bacteria in the foods that you eat. Okay. So in addition to helping converting pepsinogen into pepsin, it's also a factor in controlling the bacteria. There are some bacteria, regardless, that can survive in the acidic environment, called Helicobacter pylori, which is oftentimes associated with peptic ulcers, because Helicobacter pylori is a corkscrew-shaped bacteria that breaks through the lining, the mucus lining. So by the way, the hydrochloric acid in your stomach creates like a pH of 1, less than 1. That's acidic enough to burn through metal. So the reason why it doesn't burn through your stomach is because you have that lovely mucosa layer that produces that mucus. So the mucus kind of is what protects your stomach lining. So when you have the Helicobacter pylori, what it can do is that it can break through the mucosa lining, and then it digs its way all the way through the submucosa to the muscularis where it hangs out. When it does that, it creates an opening, so the acid that's produced can make its way through those linings to the muscularis lining and then cause ulcers. The other reason why people would have peptic ulcers is because they take non-steroidal anti-inflammatory drugs, or NSAIDs, and that impacts the ability to produce that mucus lining. So it causes that mucus lining to become thin, and then the acid that you produce in your stomach eats away until you have the ulcers. So there's kind of 2 reasons for peptic ulcers. All right, but hopefully that will help you remember that there is hydrochloric acid in your stomach, and that's what it does, and that's what happens to human health should that protective stomach lining disappear. Okay. All right. So that's the hydrochloric acid. Then you also have, we didn't really talk about this yet. There is an enzyme that's actually produced in the mouth, but it doesn't do anything in the mouth until it gets to the stomach, because it has to have an acidic environment to activate it. Which enzyme am I talking about?
The salivary lipase?
Yes. Excellent. So, and it's close, salivary is close, but it's lingual lipase. But I can see why you would say salivary, because it's produced by the salivary glands. So the lingual lipase becomes activated in the stomach. Now, in addition to the lingual lipase, you also have the gastric lipase. And both of these do a minimum, it kind of begins the breakdown of lipids. Doesn't do a lot, but it does a little bit. And that's why, we really associate lipid breakdown occurring in the small intestine, because that's where the main ones are being done. But because there's minor breakdown, it's going to impact what part of my candy.
The fat.
Coconut.
Yes, the coconut or the fat. Yep, exactly. All right, so that's what happens in the stomach. Okay, then let's move down to the small intestine. Small intestine is fun. Lots of things happen here. Okay, so what happens here? We know digestion. Okay. And the main part, the digestion happens in what segment of the small intestine mainly?
The duodenum?
Perfect. Why?
Because it's when it starts breaking down the lipids and the fat into their. So it breaks out the polypeptides to dipeptides, and then from that they can go to final digestion.
Does the small intestine produce those chemicals to do that?
No, but the pancreas and the
There
liver
you
and the
go.
gallbladder all dump it into the duodenum. So it
That's
becomes this like
exactly why.
magic poison in there.
Yeah, exactly. Okay. So the reason why it's mainly in the duodenum is because it connects. It has ducts that connect to the sensory organs, specifically the pancreas, the liver, pancreas, liver, and gallbladder. Okay, just like you said, which means we have a bunch of chemicals we have to go through, right? So there's chemical digestion happening here. And let's list them off. So let's start off with secretions of the pancreas. What does the pancreas dump in here?
Pancreatic lipase. How do you pronounce it? Lipase or lipase?
Lipase.
Lipase. Lipase.
Yep.
Okay.
So let's do pancreatic lipase. And what does that break down?
Lipids.
Yes, ma'am. Okay. Keep going.
Okay. Then for the proteins, you're gonna have the trypsinogen, the chymotrypsinogen, I think.
Chymotrypsinogen, yep. Chymotrypsinogen, okay.
And there was the other one that's carboxypeptidase or something.
Excellent. You can do perfect on the exam. Pro-carboxypeptidase. Carboxypeptidase.
Carboxypeptidase. Okay.
Okay. So what do you notice about these chemicals?
They're all inactive.
Yes, beautiful. You do not want to activate these in the pancreas. Okay, so if these ever get activated in the pancreas, it's bad problems because it's going to start to break down the pancreas itself. Okay, so that's why these chemicals are always in the inactive form until it gets into the small intestine, where then it has the chemicals to activate them into trypsin, chymotrypsin, carboxypeptidase. Okay, there are certain cases where, because of blockage issues, the chemicals in the small intestine backflows into the pancreas and activates this. And when that happens, it's going to start to eat the pancreas down, and that's going to cause, very high mortality. Because, if you have pancreatic cancer or if you have pancreatic breakdown, you can die from that. Okay. So that's why it's really important to understand that these are all inactive in the pancreas. It does not get activated until it reaches the small intestine. Okay? But once it does break down proteins. But now proteins have already been broken down into peptides, right? So technically it breaks down peptides further. Okay, so maybe the polypeptides into peptides, a little bit of the peptides into what? Amino acids. Okay. All right, keep going. What other chemicals does the pancreas produce?
The bicarbonate.
The bicarbonate is actually something that is made in the small intestine because it's receiving the chyme. So the food that's broken down in the mouth is called bolus. The food that's broken down in the stomach is called chyme. And that chyme is very acidic, right? Because of the hydrochloric acid. So that's why in the small intestine, it produces the bicarbonates to neutralize the acidity of the chyme. so that it doesn't it's not too acidic in the small intestine.
So it's not produced by the pancreas then?
No.
Oh, okay.
Yeah.
Change my notes.
Let's see. What else though?
The carbohydrate one, which is the pancreatic amylase.
That's right. Pancreatic amylase, which breaks down what?
Carbs.
Yep. And one more.
Is it sodium bicarbonate?
So that it is sodium. So sorry, I had to double check my memory. So you were right. The sodium bicarbonate is produced in the pancreas, but we were talking about that earlier. But there's still one more left. So don't change your notes. So you've got breakdown of lipids, breakdown of carbohydrates, breakdown of protein. What else have you not broken down? You also have the RNase and the DNase that's produced in the pancreas that breaks down the nucleic acids. Okay. So those are the breakdown chemicals. Then you're right. You do have the sodium bicarbonate. So you were right. Sodium bicarbonate that helps to neutralize. It doesn't do any breakdown though. It helps to neutralize the acidity. Neutralize acidity of stomach. Okay. All right. Questions? Okay. All right. So let's move on to the liver. What does the liver do?
Produces bile?
Yes. So the liver produces the bile. And what does the bile do?
Helps break down lipids.
Yes. So it emulsifies the lipids into micelles. Okay.
Is emulsification like breaking down? Is that what that means? Emulsifies?
So there is a distinction between the terms. So when we break things down, we actually like separate bonds. Where emulsify is just to separate. There's no bonds holding them together, but they're just kind of like stuck together. So emulsify is like putting it into its smaller parts. Like so for example, you have glucose being held together by specific, bond chains, chemical bonds that hold them together. So then the enzyme amylase breaks that chain down. So that's the actual breakdown. Where with fats, the goblet of fats, they just tend to stick together. So there's not an actual chemical bond that holds them together. So when you emulsify them, you just kind of like separate them. Do you see the difference?
Okay. Yes.
Yeah.
Thank you.
Yep. Okay. So bile emulsifies lipids into micelles. And what else? That's good. And then you have the gallbladder. What does the gallbladder do?
It stores the bile.
Yep. And not only does it store the bile, but it does what else?
Transports it to the small intestine.
It does, but it also concentrates the bile.
Mm.
Concentrates the bile. Okay, so it makes it a lot dense. It sucks out the water, so it becomes really concentrated. Okay. So can you live without your gallbladder?
Yes.
You can. Absolutely. It's just when, you have to be careful, monitor more of the lipids that you ingest, because the bile that you produce is not going to be as concentrated. All right. Then another function that actually happens in the small intestine, a big one, is absorption. Okay. So absorption occurs mainly along the jejunum and the ileum. Okay. So how does absorption occur? What goes where? Describe absorption for me.
When you take the absorption of nutrients?
Yeah. So along the intestinal crypts, so along the villi, right? The villi creates those bumps to slow down the movement of food, increase surface area, so that you can have the final chemical digestion occurring. And the final chemical digestion occurs along the microvilli. Okay, so the microvilli has that the border enzymes that require contact digestion. So the broken down foods come into the microvilli, where it's broken down to its final parts. Like it has to be in the form of amino acids. It's got to be in the form of monosaccharides, and that happens, and then it moves through the absorptive cells into the blood vessels. Okay, so it's going to go from the nutrients, undergo final digestion, and absorbed into, let me spell absorb right, absorbed into the blood vessels. Okay, that you find kind of in the along the villi. Okay, and lipids, however, end up where? Lipids are actually,
Lactose?
yes, I love that you knew. Yeah, lactose. Okay, so different things go to different places. So the bulk of the nutrients go into the capillaries, which end up in the blood vessels, and that's transported throughout the body. And the lipids go into the lacteals. Okay, all right. Let's end that with the large intestine. Not much else happens here. So you've got absorption still. Whatever is left over is absorbed, which means mainly water and vitamins. Okay, and why vitamins? It's from the bacteria, right? From your gut bacteria. And then you have the final step, which is defecation. And voila, we're done. Questions?